Welcome to an in-depth exploration of scientific achievement. This presentation, titled, The Architect of Genomic Editing, The Life and Legacy of Emmanuelle Charpentier, chronicles the extraordinary journey of a visionary microbiologist. Together, we will trace her groundbreaking research that led to the monumental co-discovery of the CRISPR-Cas9 genetic scissors, a revolutionary technology that has fundamentally transformed biotechnology and medicine. Through visual schematics, this presentation elegantly connects the intricacies of molecular biology with the brilliance of human innovation, inviting us to celebrate a legacy that has forever changed our ability to manipulate the very code of life. Every revolutionary breakthrough starts with simple curiosity. On the left, we explore Emmanuelle Charpentier's foundational years as a microbiologist. Driven by pure scientific inquiry, she focused on gram-positive human pathogens such as Streptococcus pyogenes and Listeria, acquiring deep expertise in regulatory mechanisms, mobile genetic elements, and RNA-mediated regulation. On the right, we see how that curiosity evolved into a monumental global impact. As a pioneer in biotechnology, Charpentier co-invented the CRISPR-Cas9 genome editing technology, represented here by a molecular scalpel that modifies DNA. This groundbreaking achievement earned her the 2020 Nobel Prize in Chemistry and led her to co-found CRISPR Therapeutics, bridging the gap between an ancient bacterial immune system and life-saving human gene therapies. This map illustrates the nomadic path of discovery, chronicling Emmanuelle Charpentier's academic journey around the world from 1992 onward. Her career began in Paris at the Institut Pasteur, where she earned a PhD in antibiotic resistance. She then traveled across the Atlantic to the United States, conducting postdoctoral research on genome remodeling at institutions such as Rockefeller University, NYU, and St. Jude. After returning to Europe, she established her independent laboratory at the University of Vienna, where she discovered virulence-regulating RNAs. Her next move took her to Umeå University in Sweden, famously described as the place where her groundbreaking work on tracer RNA took place. Ultimately, her journey led her to Germany, where she founded the Max Planck Unit for the Science of Pathogens, marking a transient yet deeply impactful career. Before the advent of CRISPR, a fundamental question drove Emmanuelle Charpentier's research: how do bacteria interact with their environment and defend themselves? The following slide outlines her three-stage scientific progression. In Stage 1, defense, she investigated Listeria spp. To understand how bacteria develop and spread clinical antibiotic resistance through horizontal gene transfer. Moving to Stage 2, adaptation, she studied Streptococcus pneumoniae to explore how mobile genetic elements enable pathogens to remodel their genomes, with a focus on vancomycin resistance. Finally, in Stage 3, regulation, she transitioned to studying Streptococcus pyogenes, examining how small non-coding RNA molecules regulate the synthesis of virulence factors. This systematic mastery of bacterial mechanisms laid the foundation for her groundbreaking genome editing discoveries. This slide presents the Umeå breakthrough, highlighting Charpentier's team's discovery of a crucial missing link in the CRISPR machinery. The pivotal insight occurred in 2011 when her lab employed differential RNA sequencing to study the genome of Streptococcus pyogenes. They uncovered a highly abundant, previously unknown transcript called trans-activating CRISPR RNA, or tracer RNA. The mechanism reveals how this molecule functions. The anti-repeat region of tracrna pairs directly with the complementary repeat sequences of the primary pre-CRRNA transcript. This pairing forms a double-stranded RNA hybrid, which is recognized and cleaved by the host enzyme RNase 3, illustrated here as molecular scissors. This process produces the mature, functional CRRNA guides that have enabled modern, targeted genome editing. This slide illustrates the multi-step mechanism of the natural bacterial immune system. It begins with viral invasion and memory, in which a bacterium integrates short spacer sequences from invading viral DNA into its own CRISPR array, creating a biological archive of past threats. Next, during transcription and maturation, this defensive array is transcribed into a long pre-CRRNA transcript. The newly discovered tracrna molecule then hybridizes with this transcript, enabling the host enzyme RNase 3 to process and slice the structure into individual, mature cRNA units. Finally, in targeted cleavage, the Cas9 endonuclease uses this dual tracer RNA, crossing RNA structure as an exact molecular guide to locate, bind, and cleanly cleave matching viral DNA, effectively neutralizing the pathogen. True innovation often requires a catalyst, the coming together of complementary minds. This slide highlights the historic transatlantic partnership between Emmanuelle Charpentier and Jennifer Doudna. Charpentier brought exceptional expertise in bacterial genetics and RNA-mediated regulation, having discovered the foundational tracer RNA maturation pathway. Doudna contributed a unique perspective grounded in RNA structural biology, biochemistry, and in vitro reconstitution, showcasing her mastery of the physical mechanics of complex RNA protein systems. Their collaboration culminated in The Synthesis, a landmark paper published in Science in 2012. Working across continents, they successfully reconstituted the DNA cleaving activity of the Cas9 enzyme in a cell-free environment, definitively demonstrating that this bacterial enzyme is guided by a specific and highly programmable tracer RNA, crossing RNA dual structure. This important slide illustrates the conceptual breakthrough that transformed a bacterial defense mechanism into a universal genome editing tool, the engineering of a molecular scalpel. Originally, the natural system relied on a complex dual RNA structure, in which separate tracer RNA and cRNA molecules needed to locate and bind to each other. However, Charpentier and Doudna's teams realized these two components could be permanently fused to create a single chimeric molecule. The outcome of this innovation is the engineered single-guide RNA, sgRNA. By simplifying the system from a two-molecule complex to a single, streamlined guide, the researchers developed a highly efficient molecular toolkit for surgery. Now, by merely altering the sequence at the targeting end of the sgRNA, scientists can direct Cas9 to precisely cut and edit virtually any DNA sequence. This timeline outlines the remarkable decade-long journey of CRISPR as it evolved from a basic laboratory tool into life-saving human therapeutics. The journey began in 2013 with its institutional foundation when Jennifer Doudna and Emmanuelle Charpentier co-founded CRISPR Therapeutics and Genomics to commercialize this groundbreaking technology for biomedical applications. Growth accelerated in 2015 with a strategic alliance, as CRISPR Therapeutics partnered with Vertex Pharmaceuticals to directly address the underlying genetic causes of severe human diseases. This effort reached a significant milestone in 2023 when the UK MHRA and US FDA officially authorized CASGEVY, the world's first CRISPR-Cas9 gene-edited therapy. Approved for the treatment of sickle cell disease and beta thalassemia, CASGEVY represents the fulfillment of the ultimate promise of rewriting medicine. As CRISPR transformed medicine, it also created a complex landscape of intellectual property issues. At the heart of this situation is the dispute, an ongoing patent conflict that has lasted for decades between the CVC Coalition, Charpentier, Vienna, California, and the Broad Institute, which centers on the priority of CRISPR's use in eukaryotic cells. This legal battle has manifested globally along geographic lines. In the United States, the Patent Trial and Appeal Board, PTAB, ruled in favor of the Broad Institute for Eukaryotic Applications. However, CVC still holds over 60 U.S. patents that cover all cell types. In contrast, the European Patent Office tends to favor CVC due to specific priority filing dates. This divide impacts today's market reality. To operate globally without legal risks, commercial entities must navigate a fragmented market, requiring them to obtain dual non-exclusive licenses from both the Broad Institute and CVC, Genomics. This slide presents the pinnacle of recognition, which organizes Emmanuelle Charpentier's numerous accolades into concentric tiers of global achievement. The outer rings represent her extensive societal impact and affiliations with various academies. Her influence extends far beyond the laboratory, highlighted by her inclusion in the Time 100 Most Influential People, her appointment as a commander of the French Legion of Honor, and her induction into the National Inventors Hall of Fame. The core layers signify her outstanding scientific excellence. She has been honored with some of the industry's most prestigious awards, including the Breakthrough Prize in Life Sciences, the Wolf Prize in Medicine, the CASGEVY Prize in Nanoscience, and the Japan Prize. Together, these honors celebrate a transformative career that bridges profound fundamental research with significant global impact. True visionaries go beyond individual discoveries, they create institutions to advance their work. This slide highlights Emmanuelle Charpentier as the institution builder, and focuses on the Max Planck Unit for the Science of Pathogens, MPUSP, which she founded in 2018. The mission of the institute is to unravel the complexities of pathogens and their interactions with the environment through unrestricted fundamental research. The institute is structured around three key areas. At its foundation is interdisciplinary research, which drives advancements in omics, genetics, and structural biology. Supporting this foundation are two core pillars: a bioinformatics platform that offers customized genome and transcriptome analysis, and a proteomics platform dedicated to profiling pathogen protein expression. Together, these elements create a strategic blueprint for future biological breakthroughs. True science is always advancing, and the discovery of SARS-9 was just the beginning of a vast research frontier. This overview outlines 6 key areas that are currently driving Charpentier's laboratory forward. First, we explore the microbiome and pathogenesis, specifically how L. plantarum contributes to enteric infections. Next, we examine translation regulation, focusing on YEBC's role in preventing ribosome stalling. Additionally, we investigate alternative CRISPR systems, highlighting type 5A metal coordination. We also look into flagellar biophysics as it relates to Salmonella motility. Finally, we spotlight epitranscriptomics by examining A to I RNA editing dynamics and CRISPR taxonomy, which maps rare nuclease variants to trace evolutionary relationships. Collectively, these areas demonstrate that rewriting the code of life was merely the beginning of uncovering nature's deepest secrets. We conclude our journey with a powerful reflection on identity and ethical stewardship. Emmanuelle Charpentier's own words anchor this final chapter. "I haven't changed, and I won't change. The scientist that I am got me here, and that is the scientist that I want to remain." Despite her monumental global fame, her core commitment is still focused on pure discovery. However, with historic innovations come profound responsibilities. Charpentier actively advocates for the ethical use of genetic technologies, including co-authoring the 2019 global call for a moratorium on heritable human genome editing. For her, engaging in the intense ethical debate surrounding CRISPR is not a distraction. Rather, it represents a crucial extension of molecular biology's long-standing responsibility to humanity. This final slide effectively concludes our retrospective by providing a thorough overview of the bibliography that highlights Emmanuelle Charpentier's legacy. It features 8 essential peer-reviewed articles that trace her scientific journey, starting from her early microbiology research in 1998 to her groundbreaking CRISPR discoveries between 2011 and 2015. In addition to these key articles, the bibliography includes valuable biographical accounts and historical reviews from reputable sources like Nature and the Nobel Foundation. Together, these carefully chosen references lay a strong foundation for our presentation and demonstrate that her revolutionary impact on biotechnology and medicine is deeply rooted in rigorous, peer-reviewed research.